Thank you very much, Kathleen. And for the sake of time, I propose to move on to the last presentation, and we will leave the questions for discussion at the end. So I would like to think further about the possible applications of study of liquid biopsies in a clinical setting. What can we learn, and what are the questions we can ask? So are we ready to really use these techniques in a clinical setting? So we've already heard a lot about liquid biopsies, what these are. I like to remember that the word biopsy is in fact from the old Greek word bios and opsis, looking at life. It's just a nice thought to think what we're actually doing. And I would like to concentrate in my talk further on the use of the study of cell-free DNA or circulating tumor DNA. So, of course, as we already heard, in the context of neuroblastoma, it can be really interesting to study circulating tumor DNA, which is a fraction of the cfDNA, a variable fraction, as we have heard, depending on different factors that we don't really know yet, probably linked to disease status, the disease load, the degree of necrosis of the tumor in place. And in the general workflow, when we're studying cfDNA, what we are currently doing in many labs throughout the world is collecting plasma samples at different time points at the diagnosis or later during disease course, getting various volumes. We can work with volumes as low as 100 microliters. We can collect these samples on what has been done in the past, EDTA, or the more recent, more specific tubes that might have certain advantages. And what is important is the centrifugation to really separate the cells from the plasma immediately, store the plasma, and then make this available for a cfDNA extraction that can be used for commercialized kits. And what we want to really do when we look at cfDNA is, as already mentioned earlier in the talk, get profiles where we have these small DNA fragments, around 200 base pairs, and not the contamination of genomic DNA that's coming from the cells. And so what can we use this for? Well, first we can use this for molecular diagnosis and looking at what we would look at for molecular markers in a diagnostic setting for neuroblastoma patients. In this context, I want to mention, in fact, that there has been data out there for the use of ctDNA in neuroblastoma for over 10 or 15 years, because in fact several collaborators have used ctDNA for detection of MYC-N amplification. And this was work done by Valérie Combaret and also colleagues from Japan, who could clearly show that we can detect MYC-N amplification in the ctDNA with a high degree of specificity and sensitivity. Furthermore, we can also use these samples of ctDNA to obtain the copy number profiles. This morning we heard about the importance of copy number profiles, especially when we're talking about adaptation of treatment, looking for profiles according to numerical versus segmental alterations. And we have shown that we can readily use these plasma samples and cfDNA to look for copy number alterations. We can do this using a commercialized OncoScan platform, because this is made for small fragments, initially for FFPE, so this works very well for the cfDNA. And in this work that was published over a year ago, we could show that this is feasible. However, we do see cases with heterogeneity. I want to insist on this, because this is important in a clinical setting. We have cases where we see certain copy number alterations only in the cfDNA and not in the tumor, or the other way around. So it will be important to think about how we take this into account if we want to stratify treatment according to alterations detected in cfDNA. And then there are other techniques, as Brent mentioned briefly, where we can actually look at the copy number alterations, also using a low coverage whole genome sequencing, just to show that this gives really nice profiles that can be readily interpreted. We can also use the cfDNA to look for mutations. We have heard about this, and a recent work has also shown that just looking at single mutations as alt mutations can readily be done using a cfDNA, for instance, looking for alt mutations as digital droplets, PCR, as shown in a publication recently. We can use this in a context of precision medicine protocols, and we can use cfDNA on targeted sequencing panels. This is an example of a patient where we were looking for targetable alterations at the time of relapse, and we got a nice copy number profile, and also looking for mutations, and we had a DNA sample, but also circulating tumor DNA, and in this patient we could readily evidence a KRAS mutation that was present both in the tumor at the time of relapse and the metastasis and the cfDNA. However, we know that neuroblastoma is heterogeneous and that there's a high degree of spatial heterogeneity. And this nice work from Peter Ambrose's group, where different tumor samples were analyzed, insisting on the importance, in fact, on the presence of this heterogeneity. This is, again, something that we need to take into account when we're looking at these analyses of cfDNA and thinking about what results to consider for our treatment decisions. Furthermore, as we mentioned earlier today, in neuroblastoma, we also have temporal heterogeneity, clonal evolution. We know that genetic alterations change over time. We know that copy number alterations can appear at the time of relapse. We know that ALK mutations can emerge at the time of relapse, and we know that other mutations can also appear. So as we think about how to use a cfDNA in clinical applications, our question was, in fact, how we can we really get an overview of all mutations that occur during the disease course, not only using a targeted panel, but really appreciate all mutations that can happen. And for this, we thought about possible techniques to explore cfDNA. And of course, if we use targeted approaches or digital droplet PCR, we can probably get a fairly high depth of coverage, a high sensitivity, but that would be mutation specific. We need to know what we're looking for. If we want to really explore clonal evolution in a higher degree, what we want to do is get an overall appreciation. And for this, whole exome sequencing techniques or whole genome sequencing techniques are more appropriate, because that would really give an overview of all mutations that can be detected. And this is why in our lab, Mathieu Chicard and other collaborators spent some time with a bit of a fiddly step trying to adapt the standard whole exome sequencing techniques to circulating tumor DNA, and to apply a modified library construction procedure to enable to really do whole exome sequencing on ctDNA. This also requires adapted bioinformatics workflow. We're following the library construction and the sequencing. We need to, of course, have a quality control for the depth of coverage, and then we can apply the current workflow for the detection of variants and for copy number profiling. So using this technique, we can readily explore cfDNA samples by whole exome sequencing. And as shown in these examples, we can nicely depict copy number alterations. On the left is an example where we compare the copy number alterations seen in a solid tumor and the cfDNA at the time of diagnosis with concordant profiles, the overall samples. And we can also look at the SNVs seen at the time of diagnosis in the tumor and in the circulating DNA. And in this case, we looked at the cfDNA at different time points, and we could, in fact, see a mutation at the time of relapse that had not been seen previously. So when we looked at this kind of results in an overall series of 20 neuroblastoma patients for whom we had a cfDNA and tumor at the time of diagnosis, we could compare the number of SNVs detected in the tumor sample and in the cfDNA, first at the time of diagnosis. And we could see that at the time of diagnosis, there's a general overlap of the number of SNVs comparing the primary tumor and the ctDNA. However, with a number of SNVs seen only in the ctDNA or only in the tumor, highlighting again this aspect of spatial heterogeneity. For patients for whom we had sequential samples that were obtained when they were in complete remission, there were no new alterations that were detected. However, importantly, for patients for whom we had samples at a later time point and who had been in relapse or progression, a mean of 22 new mutations were seen at the time of progression, highlighting again this aspect of clonal evolution. And so what we can do then is look at the evolution of the mutated allele fractions. What we did in this experiment is look at our whole exome sequencing data on cfDNA at diagnosis and at the time of relapse, and then do a specific panel which we used for deep sequencing at all the intermediate time points. And this gives us the means to really depict what's actually going on during disease course during the treatment. And in this patient, we could in fact see that there were different clones defined according to the prevalence of the mutated allele fractions. Clones, one major clone, a minor clone, and a further clone depicted in different colors here, corresponding to the mutated allele fractions. These diminished over time as the patient underwent treatment, was in very good partial remission. And then, unfortunately, there was a relapse. However, at the time of relapse, mutations appeared that had in fact been present in a very minor clone at the time of diagnosis. And so what we can in fact infer from this data is that the chemotherapy that this patient received was efficient for eradicating this clone, characterized by a subset of mutations such as SETD7. However, it was not very efficient for getting rid of this clone with EGF1 and BRCA6 mutations, and certainly not for the red clone. So it's that kind of data that I think will be important to integrate as we try to be more efficient for treatment strategies in general and adapt to treatment depending on the clonal evolution and what happens for patients over time. What is the sensitivity? We've already heard about that. I think it's important to think about the input, as Ali also mentioned. When we're doing these whole exome sequencing experiments, we do these starting with at least 10 nanograms of cfDNA. That corresponds to about 1,000 genomes. And when we do our deep sequencing, we can detect SNVs down to 1 or even 0.1%, depending on the technologies, and copy number alterations down to 10%. So it is important to bear in mind both the input, what we've been looking at, and the output of the sequencing experiments as we move on. So we've looked at the importance of cfDNA for the aspects of clonal evolution, but we might want to also be interested in expression profiling, because what we want to know is not only what happens at a genomic level, but what actually happens at an expression level. Kathleen spoke about that in detail. And so we've thought about how to actually use the cfDNA to infer expression profiles. And there has been some nice work done on this, looking at what people call nucleosome footprinting. So what is this about? In fact, this is starting from the observation that the little fragments that are in the blood, the cfDNA fragments, are of a size of about 170 base pairs. So that actually corresponds to the length of a DNA wrapped around a nucleosome. So people think that what we're actually seeing in the blood corresponds to a nucleosome, the DNA wrapped around this. And thus, the fragments, in fact, reflect the position of the nucleosomes. This is important because depending on whether a gene is expressed or not, the nucleosome pattern in the transcription start site is different. And so what has been described is that in highly expressed genes, the nucleosome occupation and the transcription start site is very organized and gives this kind of nucleosome depleted region. However, in unexpressed genes, this is a kind of unorganized pattern, and we have densely packed nucleosomes with less defined positioning. So as these fragments are released into the bloodstream, these will be cut by different enzymes, and these densely nucleosome depleted regions will be chopped up into smaller pieces, and we will result, when we look at sequencing data, at a very low coverage, whereas the densely packed nucleosomes will result in a higher coverage. So we can infer the occupation of the nucleosomes in a transcription start site just based on the occupation and then the coverage in sequencing data. So that has been published in some adult cancers, and we sought to implement that in data from neuroblastoma patients. And we did not want to do this on whole genome sequencing data because for cost issues and practicalities. So we developed a capture design looking at transcription start sites of over 34,000 genes. And what we wanted to do was then see if we can infer the expression pattern based on the nucleosome occupation. And we first tested this in a model looking at cfDNA from a healthy donor, and then in patient samples from neuroblastoma patients. And what we can see, and you can look at further details of this in a poster, is that we can in fact indeed see different coverages based on this nucleosome footprinting. And then we can look at the differentially expressed genes. And for instance, in genes that are expected to be highly expressed and low expressed, in our model, in the test case, we get this nice difference. These are the non-expressed genes and the highly expressed genes. And then we can look specifically at our samples. And this is an example looking at TERT. And we have one case where we get this kind of curve where we infer that in this case TERT is not expressed versus another case with TERT expression. So we think that this data will be complementary to the DNA analysis that can be done. So that is looking at the overall evolution, but how can we use cfDNA to analyze tumor burden and in the context of MRD and surveillance? First of all, as has already been mentioned, we know that the cfDNA quantity in fact correlates with tumor stage. Higher stage, of course, release higher quantities of cfDNA. And as we move towards higher precision and more precise quantitative markers, I think if we're really looking at techniques for MRD and minimal disease detection, digital droplet PCR looking at specific markers is probably the technique that should be adapted for this. And I would like to mention this very nice publication of our colleagues of Angelika Eckert's group, where the MIC-N status was studied by a digital droplet PCR, and the amplification could readily be identified in these experiments. So I hope that I have shown you that we have really a lot of evidence that cfDNA and liquid biopsies are really of usefulness, both to study the mutational profile, genomic information at given time points that can be of importance, especially in the context of precision medicine. We can use this to study clonal evolution. We can also use this probably to infer expression profiles, and further studies are needing to be done if we want to use this for the tumor load or MRD. I think we all agree on the importance of sample collections now, and I think there can be a general consensus statement that especially in the context of early clinical trials, there should always be sample collections to enable these ancillary studies so that we can have these important sample collections based on the SOPs that are currently being elaborated in different settings so that we can have the samples ready for the studies we want to perform. And I like to think about it in this way as we look at different situations, looking at the patient who's going through at the diagnosis, treatment, follow-up, and relapse. We can either use these techniques for molecular characterization at the time of diagnosis, for treatment stratification, as we discussed also this morning, looking at ctDNA to study CFA, SVs, SNVs, the expression levels, other potentially non-genetic alterations, the methylation profile, and at the time of relapse in the context of precision medicine. On the other hand, as we're looking at MRD and overall disease burden, this requires other techniques, and we need to put more effort into the search for the ideal marker based on the sensitivity and specificity. I think currently there are a number of clinical trials that are already incorporating these kind of studies, and a number of biobanking protocols that are based on additional sample collections in SIOP-N. Future studies will also be, of course, focus on sample collections as it's being done in a COG currently. Therapeutic trials at the time of diagnosis will be based on MRD evaluation, and at time of the relapse, a few studies are already looking at ctDNA during the disease course in the context of ancillary studies based on these programs. However, I think that for the MRD question, as has been discussed with many colleagues, there's a need for comparison of different techniques, and we need to move further into the determination of the sensitivities, because what we will want to do in the future is compare imaging approaches, pathology, including immune cytology, immunohistochemistry, the mRNA detection that we have not had time to talk about here, but I think there are a number of very exciting presentations during the coming days, and integrating the ctDNA studies that I mentioned. So I think there's still a lot of work to be done before we actually can decide what techniques to integrate and what kind of clinical studies. Thank you.
Hi, can I just ask a question, or comment and question to Kathleen? So just for those RNA skeptics, if there are any, because RNA, microRNAs and mRNAs can be very stable if they're collected appropriately, just stored appropriately. I think that's enough.
Yeah. Kathleen, do you want to come up and comment on that?
Yeah. So just so people realize it is possible to do these studies reliably and reproducibly if you process your samples correctly.
Yeah, and if just you have to keep in records on how you process your samples.
Yeah, yeah.
And just knowing that, how you process it, is already important.
Sure. And then a question about your microRNA signature. I think you said your 9 microRNA signature was identified in serum, is that right?
Yeah.
And did it validate in plasma?
We didn't test yet.
So that's an interesting, I don't know, dilemma question, given the differences between serum and plasma. I mean, I completely agree with your comments about plasma being more reliable, I think, and
for
more
mRNA
reproducible.
at least. So we see a reproducibility. For microRNAs, we see a good reproducibility. So I expect to have similar, that this microRNA signature will also work in plasma. But we have to test, of course.
Sure.
Yeah.
Sure.
But for mRNA, plasma is better. Yeah.
Thanks.
Any other questions? So can I challenge the audience? Imagine a patient in relapse. We are thinking about putting this patient on a trial with targeted treatment. We have not been able to sample any primary tumor because there was no primary tumor and there was no bone marrow. There's just MIBG spots. So we've got a plasma sample and we're looking for an ALK mutation, and we detect an ALK mutation in the plasma. Would you agree to base your ALK targeted treatment on this kind of result? Yeah.
I mean, as a clinician, yes, absolutely.
Yeah.
You said a clinical trial,
Yes.
so if the clinical trial was written so that
Yeah.
that was
So-
an eligibility criterion,
Yes.
sure. But I mean, we've been impressed with the quality of the Foundation Medicine data, and as part of clinical practice, we are acting on actionable cell-free DNA lesions.
Yeah, so I think the comment after that is that currently most clinical trials are not written in that way, and that many early clinical trials that are biomarker oriented are based on tumor tissue analysis and not ctDNA analysis, and maybe that is one aspect to take into account for early clinical trials, because I think we have enough robust data now when we're looking at mutation profiles.
I would still ask or consider what was the frequency of ALK mutation. I mean, it's different if you find it in 10% of the sequences or if you find it in 50% of the sequences.
Yeah, sort of.
Otherwise,
Yeah.
That would be my question as well.
Yeah. But then,
I mean,
yeah.
Yeah,
Be
I mean,
correct
we're
for
all
that.
dealing, inference from the cell-free DNA is that it's a subclonal mutation,
Yeah.
and is not the driver. That's a different story.
So
for
the
sure.
first challenge is knowing the ctDNA content in the cfDNA fraction, and once we know that, looking at the ALK mutation. Yes, Levin.
Ideally, you would want to know how much percentage of the circulating DNA is tumor-derived, and then you can calculate
Yeah,
the
exactly.
yeah.
That's
Okay,
what I,
yeah.
yeah, what is it? Yeah. Okay. Okay, any other questions? So I think we are all tired after a long day. Thank you for staying to the end and for listening, and have a nice evening.